We are here at CPHI Japan 2015. I'm here with Thomas from the company KBI Pharma. Thomas, how's the event going for you so far?
Great. Well, first of all, Scott, I'd really like to thank you and the rest of the UBM team for putting on another great event, and I'm really pleased with it and looking forward to exhibiting again next year.
That's great. So I met Thomas last year at the show. Since then, Thomas has shown me around Tokyo because this is my second time, and Thomas has been great. He's actually got, as you'll see, in English and also Japanese, and I've just picked up one of his business cards, also in English and Japanese. So it's very important when you're coming to the Japanese market. Thomas, I hear you have some new developments happening within KBI Pharma. Is that correct?
That's right. Yeah.
Okay.
Actually, last year we acquired a new facility from Merck in Boulder, Colorado, and this is a GMP fermentation facility that gives us late-stage and commercial manufacturing of recombinant proteins in E. coli up to 1,500 liter scale. So we're very excited, and we're already working with clients and busy manufacturing.
Okay, excellent. And what does that mean for the future for KBI? What are you looking to achieve by that acquisition?
Well, that gives us an important capability in fermentation, the large scale. Also brings us into the late stage and commercial manufacturing, which we didn't have up until that point.
Okay.
So it's a very nice complement to the cell culture manufacturing that we have at our Durham site.
Okay, great. And the feedback on the floor that something happened with mass spec, is that right? Someone was saying that KBI mass spec?
Yeah, thanks. Well, KBI has established a mass spec core facility several years ago. We've got very state-of-the-art technologies, the Water Zevo G2 and G2 second generation instruments. Gives us fantastic capabilities for characterizing monoclonal antibodies and other recombinant proteins. And as an example, one of the things that we did recently for a client, bispecific antibodies are very important in the therapeutic antibody field. So we saw a need for a sensitive method to determine what's called the heterodimer purity level in the bispecific antibody. The protein itself there's very small mass differences between the heterodimer and the homodimer. So we were able to develop a very sensitive and efficient method for rapidly characterizing and quantifying the heterodimer content in a bispecific antibody.
Okay, sounds very interesting. And over the last 1 year, since I've met you at this show, you were obviously an exhibitor last year, and you've decided to rebook and exhibit again. What are the changes that are taking place within the Japanese market that you've noticed?
Well, it seems like biologics, the interest in biologics, both innovator molecules and biosimilars, is increasing as time goes on. I've been, exhibiting with CPHI Japan for, I think maybe 5 or 6 years.
Yep.
And each year it seems like the interest level and the number of companies that are jumping into the antibody field is increasing. So that's a very positive development, and it plays well into KBI's strengths.
Okay, excellent. And why do you believe that is? I mean, I hear that a lot on the floor with the exhibitors. They're saying that is the case, but what's behind that? What's the reasoning?
Well, I mean, biologics in general are a much more important part of the new drugs under development, and therapeutic antibodies is one of the major classes of new drugs. So I think that it's just a general trend in the industry, and Japan is really focused a lot more on biologics than maybe in previous years.
Okay, and you can also help some of these Japanese companies with is it particle characterization? Is that right?
Exactly. Exactly. One of the issues with pharmaceutical formulation is if there's particles which can be subvisible or visible particles that are in a liquid injectable that's given to a patient, this can cause real problems for a patient. So the regulatory agencies are very keen on characterizing and identifying, quantifying particulates. So there's a number of methods that we've used in the past, like HIAC and microflow imaging, but we've advanced and we've established a particle characterization core facility and brought in industry experts from Amgen in particular, and brought in new technologies. So we have virtually all of the latest technologies, including Archimedes and the morphology GID G3 system. So we can basically run all of the complementary and orthogonal techniques for particle characterization within the KBI core facility.
Okay. Okay, excellent. And your plans within, I mean, as a company, KBI, what are your plans for the next, say, 1 to 5 years?
Well, I guess we'll continue doing more of the same. We continue to add capabilities. We've added another cell culture, GMP cell culture manufacturing train. So we're booking up our capacity. Hopefully we'll be able to add additional capacity there.
Okay.
We'll build on the new fermentation capacity. We'll continue to introduce new technologies and the latest developments and apply those to antibodies as well as other recombinant proteins. Another example of something that we've really developed over the past several years is the Amber microbioreactor system, which is very miniaturized, 10 to 15 ml, essentially mini stirred tanks, and we use these in process development, and we've actually been able to demonstrate scalability from these 10 to 15 ml culture vessels up to 2,000 liter production vessels. So it gives us a very powerful tool to reduce timelines and increase efficiencies in the manufacturing process.
Okay, great. Well, you've heard it. If you want to come down and learn about KBI and obviously the expertise that they offer, come meet Tom and the team here. CPHI Japan 2015.
Great. Thanks, Scott.
Thanks, Tom.